Okay, so this is going to be a short video. Let's go over symmetry and um, germ layers. Okay, so symmetry, let's talk about the different organisms that you need to know that uh, are structured in these different symmetrical patterns. Number one, you have no symmetry, which are the sponges at the periphery. And since sponges are vaguely, some of these do have some shapes to them that can be kind of vaguely defined as symmetrical. By and large, they act as single cells that are kind of connect to each other through this like extracellular matrix of spicules and spungen and whatnot. And they don't really act like a single organism. Usually, when we refer to symmetry, referring to a specific body plan where individual cells come together to form a cohesive unit, right? The sponges don't act like that, so they don't have any symmetry. Next, we'll go to the radial symmetric. The symmetric organisms. These are the cnidarians, the anemones, the hydra, whatnot, the jellyfish, and the ctenophores. And we'll do a top view and a side view. Top view and side view. So radial symmetric means you have this axis, so this axis here is coming out of the screen towards you, and the same axis is pointing upwards. So there's a top view, there's a side view. This axis, you can rotate different cuts, rotating around this axis in each individual cut. So we'll cut this way, or we'll cut this way, or we'll cut this way, which will refer to cuts here, 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 and here. Every cut that you get along this axis will give you two identical parts, right? The side view, if you cut it like this, you get two identical parts. Okay, finally, you have the bilateral symmetric organisms, bilaterally symmetric. And these are going to be all the bilateria, which are basically all the rest of the animals that you need to be responsible for outside of the sponges and the cnidarians and the ctenophores. Okay? And bilaterally symmetric organisms, we'll use beetle for example, or a very poorly drawn beetle. Messed up on the number of legs, so we'll call it centipede. There's only one plane that you can cut through where you'll get two symmetrical or identical parts. And so let's go over the evolutionary adaptations of these different groups. So one of the themes, like we talked about so many times, is that we are trying to look at these different adaptations and see how they advance down the tree of life and how these advancements will affect and improve upon organismal function. So when you have no symmetry, again, you have no cohesiveness in this organism, you're just not a very advanced organism. You don't have collective tissues. You're just not a very organized organism. You don't act very efficiently as one single animal. Okay? The cnidarians and the ctenophores, they have radially symmetric symmetry, and by and large, they tend to be pretty non-active. Kind of sit in the floor of the ocean, wait for prey to come. The jellyfish kind of move around a little bit, but they're not particularly good at moving through their environment. That is what bilateral symmetry is good for. You have a front and you have a back, and so you can move forward or backwards in a very specific linear fashion. You become more streamlined. You're just better at moving through your environment. Additionally, what this allows you to do is it allows you to get different organs or different collections of tissues at different parts of your body. And for example, especially in terms of if you think about sensory organs like the brain, like the eyes that are connected to the brain, uh, like antennae in the front, for example, it allows you as an organism to interact with your environment a lot more dynamically. Hope that makes sense. Okay, so you can think of this as less advanced in the no symmetry, a little more advanced and radially symmetric, and most advanced in bilateral symmetry. I think there is an exception that you'll need to know about in terms of all the bilateria, where the echinoderms, like the starfish, for example, they do have radial symmetry, but when they're young, their larvae are actually bilaterally symmetric. So that'll be in a different lecture, but just know that most of these bilateria are in these later evolving groups are bilaterally symmetric. Okay, and we'll go on to germ layers. They're related to symmetry because all the well, let me go back a bit. So germ layers refer to these different groups of tissues that arise from 3 or 2 early cell types during the early embryo stage, during embryonic development. Right. So in diploblastic organisms, you'll have 2 germ layers, the endoderm and the ectoderm. And in triploblastic organisms, you'll have an extra germ layer called the mesoderm. And just as an example, the different tissues that develop from these groups, endoderm, you'll have the lining of your GI tract, right? So your entire digestive tract is layered with endodermic cells. The ectoderm, you'll have your skin and your nervous system. And your mesoderm, you'll have a lot of connective tissue, muscles, skeletal system, etc. Okay. What's important to note here is that the development of the mesoderm gives you a lot more advancement in function, right? You can imagine how all these connective tissue, muscle, and skeleton really allows you to act as an organism in a lot of different ways, right? Versus the diploblasts, which only have 2 germ cell layers, they can't do quite as much. The diploblasts that you need to know, again, we refer back to symmetry because the 2 organisms that are diploblastic are radially symmetric, at least in the organisms that you were required to know, disregarding starfish. Okay, so the diploblasts are the radially symmetric organisms, so the cnidarians and the ctenophores. The organisms that don't have any germ layers, no ploblasty, are the sponges. And the triploblasts here, refer to the bilateria. Okay, so when you put this all together, you can say that as you become more advanced, you get more germ layers and you become bilaterally symmetric. Okay, in all the ways that we talked about, you get more diversity and more advancement of function as you get these different traits. And that's reflected in the tree of life among the animals that we've looked at. And this is in your lab notebook, so refer back to this as well as the slides. So you have the sponges here. This is the common ancestor of all animals. Sponges diverge early, so they're the earliest branching organisms, quote unquote least advanced. Next, you have the cnidarians and the ctenophores, which are bilaterally symmetric, or sorry, they're radially symmetric and diploblastic. And then you have the bilateria, which are bilaterally symmetric and triploblastic. And sponges up here, no blasty and no symmetry. See, so least advanced organism, moderately advanced, and then most advanced. Cool. And that's it for this video.